Hello and welcome to my channel. I'm a medical student who is trying to do first pass for USMLE Step 1, and I thought going alone would be kind of boring. And honestly, I have checked like few videos on YouTube, which basically says that study with me. And in that video, there's a background music, slow music like lo-fi, and someone is reading their own book and asks you to read your own book at the same time. So I thought, this looks a nice idea. Why not I remix it and read the book for you? And you can you can follow me if you like, or you can just mute it and read your own book or read on the screen, as this can be act as a PDF file or online file for you, which can be accessed anywhere. YouTube is free. So let's jump right in. First few pages are about the author and the important points, the dedication notice, content. Everything is very detailed, so I just don't want to go into that, but you can read it by yourself. If you have the book, you can go through it, or you can find the PDF file online. It basically tells about the exam and the scores you require for each department. So it's quite detailed. So I just want to go and read for you. And here you can see it starts with biochemistry, then it goes to immunology, microbiology, pathology. I will be going step by step reading for you. Just bear with me. Let's go straight into biochem quite fast because I know where it is. Yeah, okay, here it is. So we have 6 topics in this. First is the molecular, cellular, then laboratory techniques, genetics, nutrition, and metabolism. We'll go through it one by one. So let's start. So this is biochemistry molecular. Here you can see the DNA, the nucleosome, the chromosome in the metaphase. Here it's the opening, the heterochrome. We'll go through all of the topics and everything mentioned over here and under below. So let's start reading. The DNA exists in condensed chromatin form to fit into the nucleus. DNA loops twice around a histone ectomer to form a nucleosome. It's like beads on a string. H1 binds to nucleosome and to linker DNA, thereby stabilizing the chromatin fiber. Phosphate groups give DNA a negative charge. The lysine and arginine give histone a positive charge. In mitosis, DNA condenses to form chromosomes. DNA and histone synthesis occurs during S phase. Mitochondria have their own DNA, which is circular and does not utilize histones. So here we have the first topic, the chromatin. It is condensed, appear dark on electron microscope. Here you can see it's dark and it's labeled in the, it says A. This is picture A. Here you can see, and the NU is nucleolus, which is over here, the bigger one. It's sterically inaccessible, thus transcriptionally inactive. Here you have increased methylation and decreased acetylation. So basically what they are saying is it's condensed. When a simple example is like when there is an headphone and it's like too much tangle, it's hard and time consuming for you to open it up. But the next one here, euchromatin, it says it's less dense. See in the picture, you can see it's quite open and appears lighter on electron microscopy. See here, it's like light on the microscopy, and it's transcriptionally active, sterically accessible, so you can use it. I was giving the headphone example. So when when the headphone is like slightly tangled, you can just easily pull it and open it up. Like it's quite easy. But when it's like really tangled, you don't want to, spend your time and just use it that way. So it's a bad example, but you get my point. What they are trying to say. Uh, in the USMLE book, there are a lot of other side notes which are given for your ease to remember a few things. You might not remember all of them, but we can try. So here is heterochromatin that is highly condensed. So HC, here is HC. So bar bodies, which is inactive X chromosome, may be visible on the periphery of the nucleus. So in the euchromatin, as I said, it's expressed, it can be used. So it's euchromatin is expressed. EU means true, that is truly transcribed. DNA methylation changes the expression of a DNA segment without changing the sequence. Involved with aging, carcinogenesis, genomic imprinting, transposable elements, repression, and inactivation of X chromosome. So DNA is methylated in imprinting. Methylation within gene promoters CpG islands typically represses, silences gene transcription. CpG methylation makes DNA mute. So methylation M and mute. Histone methylation usually causes reversible transcriptional suppression but can also cause activation depending on location of methyl group. So histone methylation mostly makes DNA mute. Histone acetylation, removal of histone's positive charge, relaxed DNA coiling, causing it to increase transcription. So as we said, if there is methylation, there is no transcription. But if there is acetylation, you have like it opens it up, like loosen it up. So this causes increased transcription. It happens when you require them. You need more DNA and more making of cells. So yes, that's when acetylation happens. Histone acetylation makes DNA active. Histone deacetylation, removal of acetyl groups, tighten DNA coiling causing decreased transcription.